Hello, my name is Sakir Ibram. I'm a third year biology major from Bridgeport, Connecticut.
My name is Talisa Moy. I'm a graduating senior integrative biology major from Charlotte, North Carolina.
My name is Kyle Ray Canada. I am a third year integrative biology major from Boston, Massachusetts.
My name is Sabria Root. I am a senior from Battle Creek, Michigan, and my major is integrative biology.
All right, the title of our project is Surveying the Myrmecofauna Fauna Across Woodland Fragments in Urban Landscapes. Ants strongly influence the functionality of ecosystems in various ways. They possess numerous mutualistic and symbiotic relationships while also filling numerous niches and acting as keystone species. The Hampton Roads region of Virginia is currently undersurveyed regarding ant diversity, distribution, and prevalence. An extensive literature review and field surveys for sample collection were conducted for 11 different sites. The effects of fragmentation and urbanization were analyzed, as well as comparisons of both native and exotic species that were found. The results of this project will establish a long-term data set that will be used as a baseline for future investigations within this region.
As mentioned before, there were 11 sites in total, 9 of those sites being from previous years. And the 2 sites from this year are Woodland Skateboard Park and Deer Park. One thing to mention about these sites, they vary in size and urbanization levels, which will show in the results. So getting to how data was collected, there were many different techniques used to collect ants, those being sifting, opportunistic, sweep netting, and using bait traps. The next step in analysis was species identification, which was mainly done by Dr. Sean T. Dash. And we tried to help him with that using compound microscopes and books for identification. The 2 books being Ant Genera of North America by Fisher and Cover, and Ants of New England by Alfred Ellison, Gotelli, and Farnsworth. The next step was then analyzing the diversity of those species collected using Shannon's Diversity Index and Simpson's Diversity Index.
For the results in Table 1, the Gosnold Hope Park and Mariners Museum, Nolan Trail have the highest species richness, as well as Hampton University Strawberry Banks had the highest abundance. In Figure 1, we see that Maramicinae was the most species-rich subfamily. In Table 2, we see Patrick Henry Mall and Buckrow Beach Park having the highest similarity of ant communities. Table 3 shows the different species that were found from each of the different sites that the ants were collected from. The top 5 species that were found were Monomorium minimum, Tetramorium immigrans, Brachyponera chinesis, Phytoli dentata, Prenopolis Impernipolis interis. We would like to discuss urbanization, which is an action of making a selected area more urban by creating more roads, buildings, parking lots, and things like that within a designated area. Table 1 talks about the abundance of ants, species richness, by using the diversity indices, which measures how close the abundance each species are to each other. This is usually due to the exotic species.
It is also important to note that urbanization leads to fragmentation, and fragmentation is when you isolate areas and it actually eventually changes the properties of the area. So things like it might cause less water in a certain area, which would make it more difficult for, say, a native species to, because that's not their natural habitat. Back to what we were saying, exotic species are species that appear in another space that is not geographically where they originate from. And native species is just the alternative to that. So there's species that are observed in a spot they originate from. It can be seen in Table 1 that urbanization plays a huge role in the biodiversity of the ants living near it. For example, HU Strawberry Banks is a location with high traffic. It has concretes and buildings in the surrounding areas. And this would affect the H and D evenness. This causes the values to be low because the exotic species that can thrive in this area dominate the native species that are not able to thrive in this area since it's not their natural habitat. The exotic species also could be competitors to these native species, making it more difficult for them to survive. We also studied another site, Mason Trail, which was another site that used for the study, but it had different results than HU Strawberry Banks. This is because although Mason Trail is a park that has some concrete, has quite a few visitors, it also has a lot of space, which allows the ants to which allows the ants room to be away from all the traffic and urbanization, makes them less disturbed, um, and basically keeps their natural environment intact. There was also another study done in Michigan and Ohio. They collected data for species richness between vacant lots, gardens, and forests. And it was also seen there that in the forest had the highest species richness, vacant lots was second, and gardens were third. I found this kind of surprising that vacant lots were second, because you would think that the concrete and all the extra urbanization might affect the ants more negatively than, say, something that was more natural, I guess you would say. But this goes to show you that fragmentation is a huge issue within ant species richness. Overall, I believe more studies should be done to get a bigger picture on what's exactly the issue is. So like, is it the high traffic that's causing the ant species richness to go down, or is it the concrete or the buildings or what have you? Some of these future studies could involve how fragmentation affect native and exotic species individually, or what exactly in fragmentation causes that.
As we close out, we would like to acknowledge Hampton University's biology department, Dr. Tolliver, and Dr. Llewellyn. We would also like to thank the NSF TIP grant. Thank you.